This video is going to quickly overview the human cerebellum and then talk about the evolution of the cerebellum in vertebrates. Some of the videos in the playlist I'll go through a little quickly, like those on the cellular interactions. And obviously, those videos go through the information more thoroughly, as does my treatment of the cerebellum for my Human Anatomy and Physiology 1 students. So when the embryonic brain forms, it has three primary vesicles. The last vesicle here, the rhombencephalon, will then give rise to three separate regions of the adult brain: the medulla, the pons, which only exist in mammals, and then the second largest part of the brain, the cerebellum. So the cerebellum is derived from this rhombencephalon. I'd like to begin with the human cerebellum, just before understanding, I think, the history of it. Just looking at its structure and its cells is important. And so the cerebellum, once again, the second largest part of the brain, behind or underneath the occipital bone. It is associated with the fourth ventricle. So there are a number of spaces in the brain through which cerebrospinal fluid passes. And so here the fourth ventricle is at the base of the cerebellum. And the cerebellum has an incredible number of cells. Now, nerves tissue is organized, and one of the most obvious places to start is the difference between white matter and gray matter. The white matter has a lighter color because it is composed of axons, and many of these axons have myelin, a lipid protein mix, which gives them a lighter color. So we look here and we see, oh, there's obviously white matter here. Old anatomists refer to this as the tree of life, or arbor vitae. But then you'll also notice that there is gray matter here as well, and that's where the neuronal soma are. And as we look at the cerebellum, much of it has incredibly increased the amount of gray matter by foliation, by forming all of these folds. So if you look at this fold, another fold, all of this gray matter. As I discuss in other parts of the brain, some of the fiber tracts composing this white matter then structurally attach the cerebellum to the rest of the brain and form the lateral border of the fourth ventricle. So there are superior cerebellar peduncles coming from the midbrain. There are middle cerebellar peduncles coming from the pons, and then inferior ones from the medulla. So fibers are reaching the cerebellum from other portions of the brain. Now, in this preparation, the axons have stained this dark blue, and so this is the white matter with the axons. And so the cerebellum is attached to the rest of the brain as these cerebellar peduncles form these tracts of axons. In the human cerebellum, there is this central unpaired region known as the vermis, named after a worm. It kind of has reminded early anatomists of a worm. But then there are these two large cerebellar hemispheres which contain most of the gray matter of the cerebellum. There is also another very small lobe called the flocculonodular lobe. And so we have multiple lobes. There is the anterior lobe, the posterior lobe, which is the largest, and then a small flocculonodular lobe. These are paired. And so here is the structure of the cerebellum, and we could name the fissures as well, which then separate these lobes. Here you can see the vermis is the wormy portion in the middle. There's the fourth ventricle, the anterior lobe, the posterior lobe, and here once again is that small flocculonodular lobe here. So here you can see those cerebellar peduncles. So there's two from the midbrain, two from the pons, two from the medulla, which anchor the the cerebellum. And so a lot of what we see is white matter. The gray matter is organized into two separate regions. Deep inside the cerebellum, there are what are known as nuclei. There are deep cerebellar nuclei. But most of the gray matter is on those folds on the exterior, the cerebellar cortex. So it is the cerebellar cortex composing most of the gray matter. The white matter is then in the interior, and you can see the highly folded nature of the cerebellum gives all of this extra room for soma that otherwise wouldn't be there. If this was just a smooth, flat surface, you wouldn't have these cells or these cells or these cells or these cells. So this folded nature gives rise to all of that. And then here you can also see that in addition to the gray matter here staining pink in the outer folds, notice that there are some deep cerebellar nuclei as well. So the white matter is on the interior and the gray matter is in two separate places. The majority of it is in these outer folds, but then there are these deep cerebellar nuclei as well. The dentate is the most highly folded and the largest. Then there's the emboliform and the globose nuclei. These are the deep cerebellar nuclei. There are three types. So the cerebellum, first off, its main function is in balance and in moderating muscle output. And so I'll be certainly talking about that a great deal. But another thing is, if you read the research, textbooks often like to summarize things. So balance, coordinating most motor function, period. The cerebellum is the second largest part of the brain, and it does seem to be functional in other aspects as well. So while those first two are very important and should be emphasized, it shouldn't be thought the cerebellum is limited to that. And so, when you come across papers with, here's a possible role for the cerebellum in learning or some other aspect, it shouldn't be dismissed, even though the cerebellum has these main roles. The cerebellum has close to half of the neurons in the brain. It's huge. And so it might have diverse roles. Quick thing, you might say, well, the motor cortex in the cerebrum told these people, hey, start taking steps, and they did. But if it was only up to them, very often the motor cortex often causes too many muscle cells to work at one time. I often refer students to, remember the first time you learned how to drive and you put on the brakes. You put on the brakes too hard. Your motor cortex said, put on the brakes, and you slammed on the brakes. You then often learn what's the appropriate number of muscle cells to use. And that's a major role of the cerebellum. It's interpreting the commands of the cerebral cortex that says take a step or walk or whatever. But then it's going to limit the muscles used to only those appropriate. And one of the ways that it can do it is look at these blue tracks here. There are ascending tracks going up the spinal cord known as spinocerebellar tracks. They're named because they start in the spinal cord, they go to the cerebellum. And they are involved in proprioception, our sense of self. So when the cerebellum is looking at what muscles do we need to take our next step, our cerebellum is measuring, are we in balance or not? All right, because if I'm standing like this and I'm going to take one step, well, I would use one set of muscles. But if I'm off balance, all right, then I would use another set of muscles. In the ear, our regions which are participating or measuring balance also then communicate with the cerebellum. So the cerebellum knows if you're off balance or not. The cerebellum knows where your body parts are. So it is in a wonderful position then to help have an input into what muscles we should be moving, what are the appropriate ones. And so, crazy thing here, but if without our cerebellum, or if someone has lesions or stroke, which affects the cerebellum, our movements are uncoordinated, we are off balance, etc. And just one last thing, I like this picture. If you look at the cerebellum, actually, the cerebellum make maps of our body, one in the anterior lobe and then two in the posterior lobes. So when we move a part of the body, or we ask, where's my elbow, where's my forearm right now, our cerebellum is mapping this. So in the cerebellum, there are little maps where the cerebellum knows, like, oh, my elbow is here because these areas are telling me about the elbow. And so that's the major roles of the cerebellum. But once again, it's big, it's complex, and a lot of it is human specific. So animal studies might not be as helpful to us. The cerebellum should not be thought of as being limited to those. And if you read the research, you shouldn't be surprised by papers that say, oh, I think the cerebellum is involved in this or involved in this, etc. So when you look at the deep cerebellar nuclei, that dentate has that neat windy folds, and so does the olivary nucleus in the brainstem. And so this kind of was the model on which the folds of the cerebellum were later developed. The outer cortex is only a couple of millimeters, and it has distinct cell layers there. So the white matter is in the middle here, that's staining blue. And here you can see all the axons. And these axons are running to these very distinct layers. So there's the white matter. The two most important ones I'd like to mention are the granule cell layer and then the Purkinje cell layer. There are also others. There are basket cells around the Purkinje cells. That's what's called the molecular layer. There's stellate cells. But I want to focus on the major ones. The granule cells, this is the densest collection of neuronal soma in the brain. Nowhere else in the brain have you packed neurons, or soma at least, as tightly. And I remember a teacher of mine said that, the question was, how many cells do we have in our brain? And he referred to a quote that said, every time I read a new research paper, there are more cells in the cerebellum than there are in the brain, which is impossible, obviously, because the cerebellum would only be a part of the brain. But every time people tried to estimate the number of cells here, they came in excess of the total estimate of the entire brain. It's now thought that almost half of the neurons in the human brain are in the granule cell layer of the cerebellum. There are so many, and they are so tightly packed. Another unique thing about the cerebellum are these Purkinje cells. These Purkinje cells, they are in a single layer. All right, they are these really big neurons. You can see how big the soma are compared to those of the granule cell. There is a single layer of cells. They're big, and their dendritic trees are flat. All right, so that instead of having dendrites that spread out in 3 dimensions, it's a 2-dimensional phenomena. So the dendritic trees are flat. That makes these Purkinje cells so very unique. Now, this lecture that I'm preparing right now is for my comparative vertebrate anatomy students. And so the interactions between the different cell types in the cerebellum, it's just a little bit beyond our abbreviated coverage. So I'm just going to refer, if you'd like any more information, you can go to these videos, because then it becomes a question of which cells are stimulating the granule cells, which ones are inhibiting, which ones are stimulating the Purkinje cells, which ones are inhibiting, where do they project to, etc. So I go into this in a bit of detail, and somewhere I have a drawing of the trees. All right. So there's all of these different cell types. I go through those. Let me just show you one more on the Purkinje cells, and then we'll move along. And so the Purkinje cells, once again, huge, one single layer, and a flattened tree of dendrites. That's a very significant feature, making them very unique. So if you look at how the dendrites would go, they would then be flat along what's called a short axis of a folium. That's one of the folds in the outer cerebellar cortex. So I have more videos on how these cells interact, but once again, just for the purpose of the lecture I'm currently involved in, I'd like to now just go through quickly how did the cerebellum evolve. So no invertebrate has a cerebellum. And when we get to the most primitive vertebrates, the jawless fish, lampreys have the most primitive cerebellum. Hagfish, however, hagfish, we could argue. I had always read that they had not. Some anatomists apparently disagree and say they have a rudimentary one. In the early classification, hagfish were thought to be a little more primitive than lampreys, which then would make sense if they didn't have a cerebellum and lampreys did. Modern classification tends to put them as equivalent branches of the vertebrate family tree, which would then imply that if a lamprey has a cerebellum, the hagfish ancestors must have had one and then maybe lost it. Anyway, the cerebellum in lampreys is very simple. It's just a ridge, just a single little ridge there. They have Purkinje-like cells, but they're not truly Purkinje cells. They don't occur in one single layer. They don't have flattened dendrites. They don't have cerebellar nuclei deep inside, and the white and the gray matter mixes. Now, there are homologies as far as some of the connections with the rest of the brain. Once again, I give a number of the tracts here. So, for example, a tectocerebellar tract is going from the tectum to the cerebellum. A cerebellotectal tract is going the opposite direction. So I give some of the details. So it is very primitive. It is less like the cerebellas of the higher vertebrates than any other, but there are homologies. So certainly there does seem to be a connection. When we get to the jawed vertebrates, and sharks are good ones to start with, one of the difficult things is we don't have the very first jawed vertebrate to study. We have modern sharks, but modern sharks vary, and they vary a lot. Look at how different the cerebellum is. So in some sharks, it's small and smooth. In other sharks, it's large and highly folded. And so, you have to be careful if you're picking a model organism to compare with, there's variation. The jawed vertebrates have a larger cerebellum, so it's becoming more important. They have an unpaired corpus and then two lateral auricles. I'll go through that in just a second. And they do have definite Purkinje cells and definite granule cells. The Purkinje cells have flattened trees. They also have Golgi cells. And so there are similarities between the shark cerebellum and that of humans. Now, the vermis is worm-like in humans, in part because of all of these folds. In sharks, it tends not to form those folds and can be smooth. So it's often referred to as the corpus instead. So the corpus of the shark is the same as the vermis in the humans. That's the second oldest part of the cerebellum, and is sometimes referred to as the spinocerebellum because it's getting input from that spinocerebellar tract, which tells the cerebellum where the body is. All right, and so that's the spinocerebellum. The oldest part, which seems to then be homologous to what the lampreys have, is that flocculonodular lobe equivalent. And so the parts of the cerebellum, they evolved in stages. The flocculonodular lobe homologue in ancestral vertebrates, the vermis homologue in the jawed vertebrates. So, you jump into the jawed vertebrates and the cerebellum, larger, more complex, a much larger role. And then it continued to evolve as we look at the bony fish, the tetrapods, and the amniotes. Now, to show why I say that, I then have to name the parts of the cerebellum, the fiber tracts of the cerebellum, the anatomy of those fiber tracts, so are they crossing the midline or not? And so this video will go through it, and right now I'm just trying to summarize that. So there are features that the osteichthyans share that the sharks lack, and then the cerebellum then continued to evolve in tetrapods, and obviously now balance and muscle movement on land different than water, arms and legs are different. So now the white and gray matter is no longer mixed, and there is a single deep cerebellar nucleus. Remember that eventually we'll get to 3 in mammals. Another cell type known as basket cells exist in all of the tetrapods. By the time you get to the amniotes, the reptiles, there is a second deep cerebellar nucleus. Once again, mammals will have 3. So the dentate and fastigial nuclei have homologs by the time we get to this point. And there are some other shared features as well. Just one other interesting feature, if you ever dissect, say, a turtle's cerebellum or in a fish, you'll notice the 4th ventricle is big. All right, so originally what it was the cerebellar cortex was a dome over a big space. And if you remember, in humans, the 4th ventricle is much smaller than that. So that was a change. You can see where in humans the 4th ventricle will relatively has a smaller size. Birds have a large cerebellum. It helps in their flight. The vermis is enlarged and foliated. And so, obviously the flight would require more complex motor commands, et cetera. So it makes sense that there are advances there. By the time we get to mammals, 2 things are happening. One, the cerebrum is getting bigger, and then the cerebrum then is going to talk to the cerebellum. Those are the 2 largest parts of the brain are going to talk to each other. And so there's this huge expansion in these cerebellar hemispheres. So once again, the flocculonodular lobe evolves first, the vermis evolves second, but now these cerebellar hemispheres get huge, and it's called the neocerebellum or the cerebrocerebellum because it's new. It evolves in mammals, and then also it's getting inputs from the cerebellum. Mammals have the same 3 cerebellar peduncles, which anchor the cerebellum from the midbrain, the pons, and the medulla. And now mammals have 3 deep cerebellar nuclei. So we see a progression, 1 in tetrapods, 2 in amniotes, 3 come mammals. And here you can see, the cerebellum is quite prominent in cats, in a pig, in the sheep. There are mammals whose relative size of the cerebellum is greater compared to the rest of the brain than it is to humans. Elephants would be notable here, but then even some apes. Apes in general have a very large cerebellum, and even though the cerebellum in humans is big, some apes have a larger cerebellum relative to the rest of the brain. So the cerebellum in mammals is important. What makes a chimp brain different from a human brain? Well, a lot of things. But the 2 big ones are, so if you were to take this chimp brain into a human brain, then 2 things. You need to make the cerebellum bigger, and you need to make the cerebrum bigger. And both by a factor of about 3. So we often focus on, our cerebrum got much bigger in evolution, but so did the cerebellum. All right, and so just, obviously I love talking about the cerebrum. The cerebrum was really important in human evolution. We shouldn't overlook the cerebellum. Its increase in relative size was comparable, about by a factor of 3. And I'd like to just end with 2 notes. One is, if we were to think of humans, what makes us different from the non-humans? A lot of things, obviously, but a couple big ones. Speech is a big one, right? So if it weren't for speech, humanity would be very different. Upright walking is another big one. That's one of the differences we have from most other apes, which can only walk upright a little bit. And the cerebellum is involved in both of these. In fact, so here's just an example. There's a mutation in a gene which will then result in speech deficits called the FOXP2 gene. And it's where it's active includes the cerebellum. So this gene changed since our split from the chimp lineage. And so the cerebellum is not the center for speech, but it appears to be involved in speech. And once again, its expanse in recent human evolution, probably then is a factor in the things which are human specific, like speech. Also, then the ability to walk upright. So there is a rare condition which involves abnormal development in both the cerebellum and the cerebrum, which allows individuals to move, but they can't walk bipedally. And so once again, in the most recent lineages, so if we consider Homo erectus here, what was the ancestral hominids, what changes were occurring to help them give rise to us? And the cerebrum certainly important, but the cerebellum as well. Also, the cerebellum, it seems has roles in even things which are very human specific, like upright walking and speech. So this was an overview of the human cerebellum and its evolution.